Hi everyone. I'm Andrew. I'm a fourth year grad student, and actually, I'm fifth year now in my Scottman's lab. So I'm going to tell you about multiplexed mapping of 3D organization of regulatory proteins within the nucleus. And so we're going to start with my favorite topic, gene expression. And so it fascinates me how one genome can give rise to every cell type in the body, and each cell type has its own transcriptional program. And so traditionally, we think of this in linear space, with basically which proteins are binding where in the genome and when to regulate genes X, Y, and Z. But in the past decade or more, it's become clear that 3D organization is playing a role in gene expression. And so our lab has been developing a toolkit to study this. And so we're developing a toolkit to study multi-way protein interactions in 3D space with transcription and eventually in single cells. So I'm going to tell you a little bit about our journey there and where we're at. So to do 3D structure, our lab developed Sprite. Oops, come on. Oops. Then we wanted to incorporate transcription, so we incorporated RNA into the Sprite pipeline. And then the most elusive so far has been multiplexing protein interactions. And so I'm going to tell you how our journey of multiplexing protein interactions, what we're doing with it, and how we're applying it to 3D structure. So looking at protein interactions is quite a daunting task. And that's because in the body, you have 200 different cell types, and each cell type has its own kind of epigenomes where the proteins are expressed differently depending on the cell type, and the number of proteins that you need to study to get a comprehensive view of the organism is really quite daunting. So we need new methods for high throughput detection of proteins. So how do we do this? So our lab utilizes split pool, which requires that to do multiplexing. And so this requires an oligo to be ligated onto a piece of DNA. And this is a problem actually for proteins because proteins don't have nucleic acids to detect. And so we're getting around this by actually using a bead intermediate. And so we can label each bead with a oligo and an antibody and use this to detect proteins. So you can imagine that we would have a bunch of these in a lysate, but we would just get a mess of proteins and oligos and which chromatin is associated with protein. So we need to have some sort of way to distinguish these. And so to do this, we developed chip done in parallel. So it comes in two steps. The first step is oligo labeling beads and then coupling antibody to those beads. And so now we have an antibody matched with a barcoded oligo. We can do this for n number of oligo antibody pairs. And so we can do hundreds. And then we mix this with some lysate. And so we get this jumbled mess of chromatin that's pulled down with the antibody and the protein on each bead. And that's on the same bead as the oligo label. So now we need to think about how we're going to demultiplex this, deconvolute this, and match the chromatin to each protein. And to do this, step 2, we use split pool. So if we can split this pool of beads into a number of wells, we're going to use 12 wells here, and each well has a unique barcode. Add a little bit of ligase, and we can ligate on a barcode to the oligo and the chromatin associated with the protein. And so if we use 12 wells, each one has one barcode. That's a 1 in 12 probability that each bead will have the same string of barcodes. So in these experiments, we have millions of beads. So 1 in 12 is not very good. It's not enough. But if we pool it and then split it out again, these independent events will sort randomly, and we'll get a 1 in 12 squared chance of having the same string. And so ultimately, our goal is to have the probability of 2 beads having the same string of barcodes be significantly lower than the total number of beads. And so if we do this iteratively, let's say for 8 rounds of 12-fold barcoding, we can get up to 430 million different barcode combinations. And this is enough to resolve 430 million different beads in theory. So in the end, we get something that looks like this, where we have bead A with oligo A and chromatin A with the same barcode string. And this is a different barcode string than another bead in the pool. So now using this barcode string, we can link the oligo to the chromatin. And since we know what the identity of the oligo and what antibody it's associated with, and by extension, which protein it's flowing down, we can link the chromatin to a protein. So you may ask, does this work? So we did a small pilot experiment with 5 different antibodies, IgG as our negative control, CTCF to look at chromatin loops, POL2 and H3K4 trimethylation to look at active promoters, and then H3K27 trimethylation to look at silence promoters. Got a little bit of diversity in there. But IgG is clean. We don't really get any enrichment for IgG. If we compare CTCF to our ENCODE control, we see our map matches nicely across this roughly 1 megabase region. We zoom in, we can see that we're still matching the ENCODE nicely, and we're getting sharp peaks enrichment. And then if we keep going, we have POL2 and H3K4 trimethylation to denote active promoters. And then on the flip side, we have H3K27 trimethylation, and these are actually mutually exclusive in the same pool with these active promoters. So now we have this assay that we think is working nicely. So we're going to do a couple more validations here. Reproducibility across pool sizes, and does the composition of the pool matter? And so we'll look at different pool sizes first. If we use 1, 10, 35, or 50 different antibodies in a pool, the one antibody being our ENCODE control, we can see that the correlation across the genome and across this region is quite nice across all 4 of these proteins that we're using as to benchmark this assay. Similarly, if we use 2 antibodies to the same protein, for example, CTCF, we can actually, we don't see any hit in or dropout in CTCF signal. So we're not really seeing any competition effects here. So now we have this assay, we're gonna ask ourselves 3 different questions. Can we do this at scale? Can we do different types of proteins, histone modifications, transcription factors, chromatin remodelers? And can we look at different cell types? How accessible is this? How many cells do we need to do this experiment? So we're gonna start with scale. And so with scale, we did this experiment. I wasn't sure how to show this, but we have 150 different proteins in a single experiment across 230 different antibodies. And so there's 3 different categories here, and I'm going to go in and show you guys the protein diversity and what this data actually looks like for different protein classes. So we're going to start with histone modifications. And so the histone modification space is actually quite large. And so it really highlights the need to be able to do this in a high throughput manner. This is not even accounting for the different chromatin regulators that regulate each one of these histone modifications. So if we look at histone modifications and we ask ourselves, what about active chromatin? H3K4 trimethylation at promoters, or H3K36 trimethylation at active gene bodies. We can see that they're matching our ENCODE control nicely. The same is true for an enhancer chromatin, our constitutive heterochromatin, H3K9 trimethylation, H4K20 trimethylation, and lastly, polyclonal repressed chromatin, matching our H3K27 trimethylation ENCODE control, and the H2A119 ubiquitinylation. So we can do histone modifications. What about chromatin regulators? We'll look at active promoters. H3K4 trimethylation is a hallmark of active promoters, and many different proteins and complexes regulate the methylation of this promoter-bound histone modification. GR1A being a eraser, so we see that co-localized nicely with our H3K4 trimethylation, and then likewise on the other side of the coin, we have RBP5 part of the MLL1 complex co-localizing with a different promoter, NH3K4 trimethylation. So we can do active promoters. Polycomb repressed, we have our PRC1, PRC2, EED being PRC2, matching the H3K27 trimethylation, and then ring 1B being part of PRC1, matching the H2A lysine 119 cubic annulation. And then lastly, we have constitutive heterochromatin. These are read by HP1 alpha and beta. And so we can see that we're getting nice co-localization of the constitutive heterochromatin histone modification with our HP1 alpha and beta chromatin readers. Next, we look at transcription factors. Transcription factors are by far the most diverse in the nucleus. And so we're going to take a look at 6 examples of constitutive transcription factors, SP1 and USF2, stimulus response, so P53 and NRF1, and then cell type specific transcription factors. In this case, we did it in mouse VSL, so NANOG is an RFX1 are our cell type specific transcription factors. If we zoom in, we can see that we hit nice strong peaks with a very focal structure to them, and it's matching the motifs. Same with NANOG, NRF1 matching its motif, and USF2 matching its motif, localizing with this motif. And then lastly, we want to ask ourselves, ultimately, what we're trying to measure here is and understand how the cell and the nucleus regulates the loading of these RNA polymerases, mainly POL2, but we wanted to show that we can do all 3 different polymerases in the same experiment. And so if we look over here, POL1 for ribosomal DNA, we're getting enrichment with ribosomal DNA. POL2, we can do POL2 and POL3 at tRNA. So we can do, not supposed to be orange, it's supposed to be green. Anyways, we do protein diversity, but now we ask ourselves, what about cell type accessibility? How many cells do we need to do this assay? And so we titrated the number of cell equivalents down from 45 million to 5 million, 500,000 to 50,000. And so this 200, I think one megabase region for H3K4 trimethylation, we see a nice correlation, and the same is true for CTCF. If we zoom in, we can still see that these peaks are enriched, these regions are enriched, but we do notice that there is a hit in the quality of the peaks, but these are still callable peaks, and so we can go down to basically 50,000 cells. And for this experiment, we did 36 different antibodies, so 50,000 cells divided by 36 antibodies amortize the number of cells that you need per antibody to about 1,000 or 1,300, I believe. So if we look at this genome-wide, we can see that we have high correlation with all 4 of these marks that we've been using to benchmark our assay, genome-wide, regardless of the number of cells we use. So we can do different cell types. The accessibility to different rare cell populations seems feasible. So now we ask ourselves, what can we learn? And we did about 18 or 19 different histone modifications that were silated. And when we plotted the correlation of these histone modifications, we're actually a little bit surprised to see that they're all highly correlated with each other. And, it wasn't clear to us whether this made sense. Are they redundant? Histone acetylation adds a negative charge, and when you're trying to open up chromatin, you want to add a much negative charge to repel your negative DNA and open up the chromatin. Or is there some sort of code here that we can piece apart? And to try and understand if there is some sort of code here, we did non-negative matrix factorization to split it out into 5 different categories or combinations of histone modifications based on their enrichment. And so what we pulled out was active promoters, poise promoters, stress-inducible promoters, active enhancers, and poise enhancers. And I'm not going to go into examples for each one of these for time's sake, but I will show you some enhancers. And so the C4, which are active enhancers, we can see that it's at this near to 290 to 295 locus. And these are known pluripotency mirrors. And so we can see they're actively transcribed. And Nanog and these canonical C4 histone marks are here. So we have a nice example of C4 active promoters. And then we look at C5 in another region where we have, again, these active enhancer or these poise enhancers. But we also see this TEAD1 enrichment, and TEAD1 is associated with cardiomyocyte differentiation and development. And since the heart is one of the first things to grow in an organism, it makes sense that the cells will want to be poised for differentiation to cardiomyocytes. And so now we've shown you ChIP-DIP can basically enable us to do traditional studies on gene regulation at scale in a single lab. You can quickly screen antibodies. You can generate high quality maps for any cell type of interest. You can explore broad categories of regulatory proteins all at the same time. It's quick. The assay only takes less than a week, I think 3 to 4 days, depending on how rigorous you want to be. You can understand complex regulatory networks and chromatin states. And then importantly, you can plug into our other split pool based methods. And so next part, I'm going to talk a little bit about what we're doing on that front. And so just to reiterate, our goal here is to do multi-way protein interactions in space with transcription in single cells. So now we can do these sprite-based methods. And now we're adding multiplex protein interactions to this repertoire. And now we ask ourselves, how are sets of proteins regulating the genome structure? There's thousands of proteins, so we need a way to understand 3D structure and how it relates to each protein. One of the most classic examples of this is CTCF, and this has been studied to death, and we're still finding new things about it. And so this is a good example of how proteins are regulating this 3D structure, but we want to expand beyond CTCF and we want to be able to screen this very quickly. So just a recap, a reminder what Sprite is. Basically, we fragment the genome into clusters, and we have these, say we have the blue cluster and the pink cluster, and they are stably bound to each other, and things that are stably bound to each other will always sort together. And so when we do split pool, we can get a unique set of tags on each cluster. And if they're in a cluster together, we can say that they're interacting. And so we can build these interaction maps. And so in this first sprite paper, we showed that this matches high C nicely. And so we're going to integrate this in with ChIP-DIP and show that we can get 3D structure in a protein-specific manner. So we did this with 4 different proteins separately, and then we did H3K9 trimethylation, H3K4 trimethylation, H3K36 trimethylation, and CTCF. And so we can separate all these histone modifications and proteins out. We zoom in, we can see that we're getting nice structure of the 1D structure. But what about the 3D structure? And so to look at the 3D structure, we're going to look at my favorite chromosome, chromosome 19, and we're going to compare H3K36 trimethylation and H3K9 trimethylation. And you'll see a very stark contrast here. H3K9 trimethylation has quite a bit of structure to it, while H3K36 trimethylation has much less structure to it. And this is the entire chromosome. It's even more interesting when you layer on the 1D track, And we can see that it's actually enriched in the same regions of this chromosome, or close to the same regions. And it's even more interesting when you layer on where the zinc fingers on chromosome 19 are co-localized. And we can see that in the H3K9 trimethylation sample, all these zinc fingers seem to be interacting with each other. Versus H3K36, they are not. So this is an interesting observation. And we wanted to see, okay, this entire chromosome is interacting with itself at zinc fingers. Are other chromosomes interacting with itself when you at H3K9 trimethylation marks? So we collapsed the genome down into H3K9 trimethylation enriched regions, and we can see many off-diagonal examples of interactions between chromosomes, but I'm going to focus in on a single chromosome, in a single pair of chromosomes, chromosome 18 and chromosome 13. If we zoom in, we can see that that chromosome 13 and 18 are actually interacting rather robustly. And so we are excited to explore this a little bit more in depth. This is actually not a zinc finger interaction. This is independent of zinc fingers. And yeah. So next we wanted to look at CTCF, because who doesn't? And if we compare our CTCF spread IP to Hi-C data, we can see that we are getting these TAD boundaries, but the TAD boundaries are very discrete compared to these filled-in Hi-C maps. And that's because we assume because we're enriching for the actual point of contact on CTCF. And we also get these off-diagonal long-range interactions. And so we were intrigued by this, and we went looking for other examples of this. And we found one, again on chromosome 19. And if you zoom in, this is about a 7 megabase distance interaction. And so the first thing we wanted to ask ourselves, okay, yeah, this is interacting. It's got a pretty strong interaction. But are there even CTCF sites here? And so we zoomed in and used our data to look and investigate this. And we actually saw that the boundaries of these interactions have very strong CTCF sites relative to the other ones in interspersed in between. And this region is actually just littered with CTCF sites. And so to bring it all into focus again, with our goal of doing multi-way protein interactions in space with transcription and in single cells, let's see where we're at, how we're doing. So we can do 3D structure, we can do RNA with 3D structure, we can do multiplexed protein interactions. So what we still need to do is incorporate RNA into these 3D maps, protein-specific maps. We want to do high throughput of hundreds of proteins, not just 4. We want to incorporate 3D structure, and ultimately we want to be able to do this in single cells. And so this is all more to come. Stay tuned in the years to come. And so I'd like to thank everyone in Mitch's lab, especially Isabella Bronzi, who did all the analysis for ChipDip. She was actually instrumental in developing ChipDip and all that NMF data she generated. Mario and Jimmy Guo, who helped with experiments in the initial stages of ChipDip. Ben Yi, who has helped with the regulating the pipeline, the ChipDip pipeline, and of course Mitch for all his help and support. And he actually generated many of these heat maps that I showed today. And then of course, we'd like to thank all these antibody vendors that helped us get these antibodies to develop this technology. And Ina for all of these illustrations. And I'd be happy to take any questions. Thank you.